as Marisha introduced me, I am Laura. I am a bio major at Carolina University, and I'm almost a senior, so this was a very, very good experience before I graduate and go to the scientific world. Um, I was in the first 3 days boot camp that started this, so I want a little to compare what we did this week and then the 3 days before, and it was all just very awesome. So I'm gonna take you a little bit inside of what the cell culture we did inside the Xvivo and also the BSC, which is the open and closed system. So a little bit of the content we're going to see is the cell lines we used, the systems, the techniques, the pictures that we took on the microscopes, a little observations that I did between what we experienced, and of course the gallery, which has a lot of pictures. So as Walter already introduced, cell culture is a process of growing cells, but also maintaining them, which is the very purpose of the closed and open systems. So what makes the cells grow is the treatment and development of the biomedical technologies. So this project, we use the cell culture as a model to study cell behavior and assess the environmental factors that make them grow and make them allow to become what we study. So the cell culture systems that we used are the Xvivo and the Biotech cabinet. So before this camp, I did not know what a Biotech cabinet was. I've heard of it, some pictures, but I didn't have the hands-on that I had this week, which was going in, doing the protocols, having the comparison between what the Xvivo system is trying to not take out, but summarize, which is the cleaning protocols. And it was really cool to be able to compare it and be here and do this and then go to W firm and do the other thing and just going back and forth and be like, oh yeah, this is interesting, this is different here, it's different there. So that was really cool to experience and compare in real life and be like, yeah, this is awesome to experience. So the two cell lines that we used are the A549, which is long carcinoma, and the K562. So the adherent and non-adherent cell lines were the two that we mostly used between this week and the beginning of the camp. Adherent cell means that they attach to something. They attach to a surface. That's what they grow in. And the non-adherent, it means that they grow freely in the media, in the nutrients that make them grow, right? So the long carcinoma, commonly found in tissue organs, and it has a monolayer during the culture. And then the non-adherent has typically derived from blood or immune cells. And they do not require attachment, which is why the media change is really tricky to do, because if you don't do it with the protocol correctly, you take the cells out, no more babies. So cell culture techniques we used throughout this week, we kind of divided it between 2 to 3 days. The first day was Monday, we did the protocol. It was the biosafety cabinet, what to clean, what not to clean, what to use, what not to use. Because at the end of the day, it is a closed system, but the point of it is to make sure that the environment is safe for the cells, but also for us, but mostly the cells. So we did that. Well, Collins did the thawing, which is a recovered cell from the freezer, the frozen, and then warming it up and transferring them to a fresh culture media. And then we did the seeding. I had more hands-on experience this week than the last time. I think he guided me really well between what to do, what not to do, what not to break, and just transfer the cells into a new culture. I think we did the T75 flasks, if I'm not wrong. We used those, which are like I don't know how to explain it, but it's a flask. And then we did the media change on day 1. So how this is labeled the day by day is really weird. We did the thawing and seeding on day 0, and then the media change day 1. So day 1, we replaced the old culture and medium with the fresh nutrients, but we also did imaging because we have to record whatever we see, whatever we change, there's something different in the environment, we do that. So we take the imaging, which is the images on the microscope, I'm gonna see them a little later, to observe the morphology, how they grow, the confluency. I just learned that term, by the way. Didn't know what that was, which is basically how much cells you have on your flask. And you monitor them like a baby, literally. And then we did the harvesting after that on day 2. We observed the cells again. We monitor how much they grow, if some of them were dead, if there's a lot of debris, less debris. And after the harvesting, we did the cell counting. I did it manually the first time, which it was not good. It was a pain. It was a very cool thing to do because you get to see, you get to count the cells objectively, and then Collins will tell you, no, you're wrong, and you count them again. But then here we use the cell counter, the animatic one, and it was really cool to do different technologies and see how they're shaping today's science. So here are a little image of the cells. Put them here. So the ones in color are the Xvivo, and the ones in black and white are the BSC. They're not really much of a difference. The only difference will come when you get the results and you look at the viability. In the Xvivo one, I did notice that on day 0, you don't see much of a difference. They mostly look alike. They look just like little dots, like rice. And then on day 1, then you see the difference. Because you see let me see if I can put it here. They look like little shapes like, I don't know how to explain it, like embryos? Spins?
Squamous.
That makes sense. Okay, yes, that shape. And day 2, you see them more spread, or you see more cells because they keep growing each day, right? And on the BSC, they look kind of the same, but also different. There are more gaps. You see maybe less viability ones in the end. And same thing with the case 562. Difference is that the K562 shape does not change. It just stays like a rice. And some cells might die by the day 2, day 3, and that's why you keep doing the same process. You seed again, you harvest again, you cell count again, and this is just a cycle that keeps going because that's growing cells. So this was really cool to do, have both systems and compare them and see the difference, and just lively look at things that you want to see, notice things. So it was really, really cool to be able to do this and put it on paper and be like, hey, you know what, this is what I did, this is what I noticed. And this is a little of a gallery that we did. We did a lot of tours. We went to Dolly Firm, Amnion. We see this, which is like a kidney printed something. We see a lot of machines that didn't even know existed with a lot of weird names. And this is the placenta. Yeah, look like a pork chop. So if you notice, I compared things to food somehow. That works. We saw the printing. I call it chips, but they're called cassettes. If I'm not wrong, or something like that. On a chip, yeah, that you put the cells in, and you see it compared to the original shape of them. So this was all really very good to do. And in my career, which is biology, it's a lot of good things to do, but I think I'm really interested into research. So in that area, I think this is a very cool experience to be like, hey, there's a whole world outside of your career, inside of your career to do. Not just in cell biology, not just in neuro, not just in micro, just a lot of things that we can do. And it has broadened my understanding of science, my understanding of what I want to do after I graduate. I might be looking for a job, Collins. I'll call you. But it was all just exciting. Thank you to all of y'all, because this was never going to be able to be done without any of you. Thank you, Collins, for your guidance. Thank you, Dr. Jones, for bringing us here and giving us this opportunity. Thank you, Anna, for giving us a tour and a little bit of what you do day by day. And Katie also, because she helped us going to the test beds. And Ahmed, thank you for doing all of this too. And just excited because this was really, really good opportunity for us, for me too. And that's it.